Welcome back everyone to our genetics lecture series. Today we are going to be looking quite closely at the genetic code, at the process of translation, and finally at protein biosynthesis. Before we do this, I would like to recap briefly what we have learned about the replication and transcription in our last lecture. So, what are the steps of replication in a nutshell? Obviously, replication starts with the initiation. During initiations, protein bind to the DNA and open up the double helix. In addition, during this step, DNA is getting prepared for complementary base pairing. The next step, elongation, includes proteins that connect the correct sequences of nucleotides into continuous new strand of the DNA. Finally, the last step of replication is the termination, where proteins release the replication complex. So, if we look at this process more closer, then we'll notice that actually quite a lot of different enzymes are involved as well. And the first important protein in replication is helicase. And the job of helicase is to untwist the double helix and to expose complementary strands. And this is basically what you see on the right-hand side. The next important enzyme is primase, which puts down short pieces of RNA termed the primer on top of the naked DNA. This is quite important because those primers do have a free 3' end, which allows the DNA polymerase to add complementary nucleotides. And this whole site of replication is called the replication bubble. During replication, each nucleotide strand serves as a template for building a new complementary strand. Important bit here is that polymerases can only read the DNA sequence in 3' to 5' direction. However, they only synthesize in the opposite direction. They synthesize in 5' to 3' direction. Moreover, as I mentioned before, DNA polymerase requires a 3' and to initiate synthesis, and as a result, DNA strands are antiparallel. And this causes a problem for replication. So, what are the rules of replication? You need to remember that replication is semi-conservative. That means that one strand of the DNA remains unchanged and is then bound to a newly synthesized one. Therefore, the mechanism of replication is semi-conservative. It always starts at the origin, and the synthesis occurs in the 5' to 3' direction. Also, replication is semi-discontinuous due to the Okazaki fragments on the lagging strands, and replication finally requires RNA primers. So, how about transcription? In transcription, only one strand of the DNA molecule, called the template strand, is copied by the RNA polymerase, and this is an important difference if we compare this process to replication. Also, because RNA polymerase moves into the 3', 5' direction along the template strand of the DNA, the RNA product is antiparallel and complementary to the template. Lastly, RNA polymerases recognize start signals, so-called promoters, and stop signals, terminators, for each of the thousands of transcription units in the genome of an organism. Once mRNA molecule has been produced during the transcription, it needs to be processed, at least in eukaryotic cells, including human cells, because our genome not only contains coding regions, but also non-coding regions. And the name for the coding regions is an exon, and for the non-coding region, intron. So, what do we need to learn today? In today's lectures, and after watching the videos, you should be able to explain the structure of the ribosomes and the main difference between bacterial and mammalian or human ribosomes. You should also be able to explain the terms codon and anticodon, know the characteristics of the genetic code, be able to use the RNA codon table and also the RNA codon wheel, And you should be able to assign a matching amino acid to a sequence on the DNA. Starting with the DNA sequence, then mRNA sequence, tRNA, and finally the amino acid. Lastly, you should be able to understand the important steps of translation, including initiation, elongation, and termination. So, before we look at those learning outcomes, let us recapitulate what we know about the central dogma of molecular biology. I hope you remember that I already told you in one of our previous lectures that the central dogma of molecular biology means that information passes from DNA to RNA and subsequently to proteins, but it cannot pass back the information. So this is a one-way process. And today we are going to be looking very close at the genetic code, which is actually the basis for information storage on both DNA and RNA. In addition, we are going to be looking at the process of translating the information on mRNA into the growing polypeptide or protein chain. So, what is the genetic code? If we look at the definition, then we will come up with something like that the genetic code is the nucleotide-based sequence on DNA and subsequently on mRNA by transcription, which will be translated into a sequence of amino acids of the protein to be synthesized. This sounds quite complicated, but in reality it's really straightforward. So, if we look at the genetic code, then we will realize that there is a coding problem. As you remember, there are 4 bases in the DNA. We have the adenine, we have the cytosine, guanine, and thymine. So there are 4 of those bases in the DNA. However, there are at least 20 amino acids, and those amino acids are fairly different. Those amino acids are the building blocks of proteins. So, how can we now use just 4 bases to code for all of those amino acids? We have 4 bases and 20 amino acids, right? If the genetic code would be 1 base, this would allow us only 4 combinations. Obviously, that's not enough for the 20 amino acids we are looking for. If the genetic code would be coded by 2 bases, this would give us 16 combinations, which is still not enough. However, if the genetic code is coded by 3 bases, this would allow us 64 combinations. And indeed, in all cells, the genetic code is coded by 3 bases because this allows us quite a lot of combinations, at least 64. So what are the most prominent characteristics of the genetic code? First of all, the genetic code is very specific. That means that a specific codon always codes for the same amino acid. In addition, the genetic code is universal. The same code is used in all living organisms, in prokaryotes, eukaryotes, and even in viruses. Finally, the genetic code is degenerated. This means that although each codon corresponds to a single amino acid, one amino acid may have more than one codon. For example, arginine has 6 different codons. This results in 61 different codons used to code for amino acids. In addition to codons that actually lead to production of proteins because they code for amino acids, there are also start codons. And the start codon is AUG. It initiates the start of translation. What you can see here are just a few examples for codes coding for amino acids. For example, AUG codes for methionine, UCU codes for serine, and CCA codes for proline. So what you need to pay attention to here is the polarity again, because you need to start at the 5' end and end with 3' end. This is the general direction. In addition to those, We also do have stop codons or termination codons. UAA, UAG, and UGA lead to termination, stop of the translation, and it indicates finishing of protein synthesis. With this, I would like to end the introduction session, and I will see you in the session number 2, explaining to you how to use the genetic code table and the genetic code wheel. Thank you very much.